Steatotic
Okay.
acid.
Yes, but yeah,
That is the main important thing. First
yeah.
of all, gram-positive bacteria has multiple thick peptidoglycan layer, and on that layer they have the steatotic acid. And that acid binds with the crystal violet with the help of the modern. So modern acts as a bridge between the crystal violet and the steatotic acid of the peptidoglycan layer. Gram-negative bacteria, their peptidoglycan layer is very thin, only one layer, but till they cannot get color with the crystal violet. Why? Anyone can guess?
Because
It does not retain the color.
It's
It does not
contained.
retain the color because
washes away.
Why? Why? Even there is a one layer, why the color cannot stay? It can be faint. If it is one layer, it can be very faint, but crystal violet doesn't stay at all.
because of the thin wall, and it washes
Yeah,
out
because
after
it was
the
the
rinsing of the
wall.
alcohol.
That till it's only the peptidoglycan layer is responsible. Maybe in the gram-positive bacteria is staining 100%, and with the one layer it may stain 10%, but there is no stain, is washed away totally. Why?
It have no ticalic acid.
They doesn't have any tectonic acid, so that there will be no stain can stay there, because even modern iodine, whatever you did, the tectonic acid actually binds the crystal violet stain in presence of modern, and tectonic acid is present on the peptidoglycan layer. So whatever is there, there is no bridge, nothing, no tectonic acid, everything washed away. It's clear to everyone now why the gram-negative bacteria cannot keep any crystal violet stain.
The
Because it doesn't have the peptico acid.
Yes. And what modern does, the gram iodine, that is the modern, it acts as a bridge between the crystal violet and tectonic acid, so that the color can stay on the peptidoglycan layer. If there is no tectonic acid, even there is a peptidoglycan layer, what is going to happen? It cannot keep the color because modern cannot make a bridge. If any bacteria, you can see some portion is taking the crystal violet stain, and some portion or some bacteria are not in a same bacteria. They belong to each group.
Ma'am, can you repeat the question?
The question?
In a bacteria, if you see, the some portion has color and some portion doesn't take the color. So it is considered to be gram positive, negative, or what group?
Yeah, a mixed group of like they have both gram positive and gram
No,
negative.
I am not telling in a population you have a gram positive and gram negative different bacteria. In one bacteria.
Ma'am, they are called the cocobacilli.
No. Gram variable. Yesterday, remember I told you
Yeah,
guys?
gram variable. Yes, I remember.
Remember now?
Yes.
Yes, ma'am.
It depends upon, because in some portion of a bacteria in one side may be in uneven or unequal distribution of tachytic acid. This is sometimes caused due to the genetic factor mainly, and there are some other factors what we have discussed. Okay. Let us go to the acid fast stain. So we have learned about gram stain. Gram stain is the basic stain for all in general bacteria. So, so which we are going to do the grams acid first stain. Gram stain is non-reactive when some bacteria cell walls are made up of thick waxy lipid layer. And for making that waxy lipid, they are nearly waterproofed.
Drink it. Push it by yourself.
So that means in the layer, in the cell wall, they have some waxy lipid layer. If it is waxy lipid layer, what is going to happen? This gram stain is dissolved in the distilled water. These are water permeable stain we are using. But if it's a waxy lipid, you know, if there is a lipid, it's not going to take the stain, it's going to slip away. So it's waterproof. So that time gram stain is not going to work. So it's called the gram stain non-reactive. As water-based stains cannot penetrate to the cell wall to stain. What type of bacteria falls in this category? Mycobacteria, which is tuberculosis, cause TB, and leprosy. Okay, so this needs to be stained with another stain, special stain is called the acid fast stain. So acid fast stain is used to stain the meningitis, leprosy and the tuberculosis bacteria, because they have the waxy lipid layer on their cell, outer membrane of the cell. So what is the acid fast techniques? As it contains waxy lipid layer, it tells that it prevents the penetration of the water soluble dyes like the gram stain. So acid fast stains has the composition which forces the dye through the cell wall. And once the dye is inside, it is trapped in a resisting decolorization with a acid decolorizer. So with the name, you can see it is probably there is acid. So acid fast stain technique is said in microbe which resists decolorization by acid alcohol is color fast or acid fast because it maintains its color. Once it's go inside, it's trapped inside the membrane layer and it stays there. Even you cannot remove it. So most microbes without the waxy lipid layer are almost immediately decolorized by acid alcohol, which we are using acetone. And for the gram stain, we are using the acetone and alcohol. But here they are using acid and alcohol. Thus take up the color of the count stain, non-acid fast. So TB, it is the thick waxy cell wall which protects TB from disinfectant. Is protecting TB from disinfectant. So it's resistant. That's why TB is hard to clean, to disinfect. Makes TB resist drying out. It doesn't resist, it can stay for a long time in the air. Protects TB from our immune system and adds to TB's virulence. The degree of pathogenicity or availability cause disease. So what is the composition? You doesn't have to know, but this is the waxy lipid layer. It has the acyl lipid and mycolic acid over the peptidoglycan layer. So what is happening? If there is a peptidoglycan layer, even there is a techidic acid, but there is a lipid layer, waxy lipid layer. This is mycolic acid is waxy. So this layer prevents the penetration of the water soluble dye to the peptidoglycan layer. So that's why they have to choose another dye which can go penetrate through the lipid layer and get trapped here. TB needs increased containment level. You guys know you need the airborne isolation. You need the N95 mask and all the PPEs. TB cultures are processed using additional operational controls. We are not doing it. These additional controls would include separate rooms, especially designated transfer hood, biological safety hood, and which is the hood, classical biological safety hood. Number 3 or 4, where you are doing only, even you cannot, air is circulating in, it's a totally closed hood. What you have seen that one, you can put your hand from the outside through your gloves, and it should be in, or the patient or the sample should be used in the negative pressure room. So what is the primary stain? This is one of the stain. It's called the gel Nelson or ZN stain. What is this primary stain? This you have to remember, dear. Carbol fusin. This is the red. While heating, slide over boiling water for 5 minutes. Decolorizer is acid alcohol solution. Counter stain is methylene blue. Immune modification as before, but carbolfucin is mixed with phenol, which replaces heat. Oh, nowadays they mix it with the phenol so that it doesn't need the heat. So how it does, you have, probably they are going to tell you later. So basically you are going to put a small sample suspended in saline solution and fixed on a slide. You know how to make the smear, liquid smear, dry smear. Then slide 1 is flooded with carbol, fusin and phenol. That is the primary dye or first dye for 3 minutes and then gently rinsed with water. Then it's decolorized with HCl. That is the acid in 70% alcohol. That is called the acid alcohol decolorizer. So it's 3% HCl, 70% alcohol until the color appears to be removed and rinsed with water. Then slide 1. What color is removed? Not the inside color. What is trapped inside, it is going to stay color. What is the excess color outside is going to be removed. And then the slide 1 is flooded with again methylene blue, that is the counter stain, for 30 seconds and then rinse with water and air dried. So acid first stain example how it looks like. This is the TB, I guess. Yeah. So this is one example for acid first stain. What is the name of the ZN stain? That is the stain, the acid first stain, and this is used for staining of the bacteria which have the thick lipid or waxy layer. Example, leprosy and tuberculosis. Other microstains, there are a couple of other microstains can be used for staining different things. It's called the India ink. So spore stains. So we are going to, and there are fluorescent stain. Fluorescent stain is which gives, it needs a special microscope. You cannot see it on the general microscope. lab microscope. It has some special light which can help to see the fluorescence. What are the fluorescence is dye is used? Is the acridine orange. It give you the orange color, bright fluorescence color. You know what is fluorescence color looks like. So a fluorescence stain used for detection of bacteria and fungi in clinical specimen. If present, bacteria and fungi stain red orange. It is a rapid stain and that may be useful in thick specimen for which gram stain is not interpretable, because if it is too much thick stain, it's going to be hard to decolorize the stain from the background. So it is going to be hard to tell. So that is tell for the thick smear or thick slide for the grams for any bacterial, it's going to be difficult if there is a thick smear with the gram stain. Direct fluorescence antibody stain. It is specific label antibodies are used to directly detect a variety of bacteria and virus. So there is antibody has been raised specifically for that specific bacteria or specific virus, and that antibody is tagged or breached with a fluorescent stain. So when you are doing the immunostudies, when you are incubating with the antibody, with the bacteria, so what is going to happen? Bacterial antigen is going to bind with their antibody. And that antibody is tagged or breached with a fluorescent stain. So if there is a presence of any bacteria, of the specific bacteria, we are going to see the color of the bacteria. So specimens are fixed into a slide and incubated with fluorescent labeled antibody. If present, the antibodies bind to organisms and fluorescence gain under UV lights. So how it looks like? Acridine orange stain, which is the orange color, and this is the fluorescent stains with the antibody is the bright green. So counter stain is the second stain used in differential stain. This is the when you are using more than one stain is called the differential stain. Decolorizer. What is the function of the decolorizer? Used to remove primary stain from bacterial cells during a differential staining, because when you stain, it stain everywhere, including the slide, background, everything. So you have to wash the excess stain. The microbes which become decolorized are negative, and once they retain the primary stains are positive. Gram positive, bacterial cells that retain the crystal violet dye after the gram stain procedure is done is the gram positive. Gram negative, bacterial cells that take up the counter stain only saffron after the gram negative procedure is complete. So there is no crystal violet stain, the primary stain. So primary stain is the initial stain used in differential stain. Now you know. For the acid fats, what is the primary stain? We just learned. So memory is so short, we just learned.
Crystal violet.
No,
I
that is
know
the
the
gram stain.
counter
I said
stain.
the
I know the
acid,
counter stain is hydrochloric acid. I can't remember the first one.
not only the
Methylene.
hydrochloric acid and
It's the counter
alcohol.
stain. Okay.
That is the counter stain for the
Counter
acid fast
stain
stain.
for
Hydrochloric acid and the alcohol. 3% alcoholic acid and 70% alcohol.
The primary is fusing, or that's called fusing.
Carbol fusing
carbol
Yeah,
fusing.
Carbon.
That's
carbon.
the
with,
red
with,
one.
with phenol.
Carbon fusing with phenol, yeah.
You all are younger than me. Your brain should be more active, dears. You are and me in the same page in the slide. Okay, so primary stain from the gram stain is,
Carbon fusing with
Carbol
phenol.
fusing.
listen what I'm telling.
Crystal
Oh, for the
violet.
gram stain. Oh, that's
Crystal
crystal
violet.
violet.
Crystal violet.
My goodness. You have to read, you have to see whatever is the question.
Yes, ma'am.
For the gram positive, not gram positive, well, gram stain, what is the primary stain?
Crystal violet.
Crystal
Crystal
violet.
violet. Yeah.
What is the primary stain for the acid first stain?
Carbol
Carbon
fusel.
Carbol
Carbon
fusion.
fusion.
with phenol.
Okay. What is the counter stain for the gram stain?
Safranin.
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 Safranin. What is the
Safranin.
counter stain for the acid first stain?
Methylene
Hydrochloric
blue.
acid and alcohol.
Methylene blue.
Methylene blue.
Methylene blue.
Methylene blue. Methylene
Oh.
blue.
Any acid or alcohol, this is for the decolorization. It doesn't stain, dear. Do we use any mordant in the acid first stain?
No.
No. Okay.
Phenol.
What is the decolorizer in the gram stain?
Alcohol.
Acid. Yeah, alcohol.
Alcohol.
Nope.
Acetone.
No. Acetone alcohol.
Acetone alcohol.
Acetal alcohol. What is the decolorizer for acid first stain?
Acid
Alcohol.
alcohol.
Acid alcohol solution.
Acid alcohol.
I have to read all of this later.
You know the name of the bacteria which cannot take the gram positive stain, right?
Yes.
It's gram negative.
Which cannot take the gram stain. I didn't tell gram positive or negative. The bacteria cannot take the gram stain.
It's a TB.
TB and?
Okay.
Leprosy.
Leprosy.
Okay. Very carefully hear or read the question. A little bit is gram stain and gram stain and gram positive, gram negative stain is everything. You have to read it whether what they are telling, whether comparison between the different stain or comparison between the bacteria staining properties. So you can see I have already sent you all the what is called the definitions, right? For the modern antibody acid first stain gram variable. Did anyone get chance to read it yesterday? My notes?
Yes, ma'am. Yes,
Okay.
ma'am.
So maybe you have excess because that covers probably the whole unit. Okay, whole model. So you have probably have all the informations there, right or wrong? Anything missing? What about the questions here? Did you study and do the review? Yes or no?
We are being bombarded.
Who said that?
Me. So much information.
Amina, you are telling bombarded. I never heard from you that. This is strange. This is strange, Amina.
My brain is vandalized at the moment.
Okay, so unit 4 is done. Now we are going to jump up. Let us see what is left there. But this is try to make it easy. Maybe there will be more difficult one. Okay, so what's the
Brex,
video?
Dutch flower bulbs since 1818. Your best garden starts with
Enjoy
Brex
flower.
selection of
I
flower
have
bulbs,
this script.
direct from Holland. All backed with a lifetime guarantee. Visit brexbulbs.ca.
The gram stain is named after its inventor, Hans Christian Gram. As a Danish botanist and teacher of
We
pharmacology,
did that, right?
he devised
Why
this
they are
important
repeating
method
it?
of differentiating bacteria
Yes, we
in
watched this one.
1884
Yeah, we
that
see
is
this
still
one.
in
Why
common
they are
usage
repeating
today.
it? Yes. Yeah, it's useless. They are repeating it 2 times. rejection criteria, smear tense video. You want to take? Now it's a due time takes micro test, unit 1. You want to do it today?
No, ma'am. We are not ready yet.
Okay, so let us go to the unit 5.
No, miss, we are on site. No pen nor pencils.
I can tell Pera to give it to you. And you doesn't need a pen and pencil. You can go to the computer and do that. No excuse.
That is so funny. No, miss, don't do that. We're gonna do our fast test soon.
Okay. Let us go to the next chapter then. This is going to be more to remember. Amina is already bombarded. So now you have learned what is the microorganism, mainly the bacteria, how to stain them, how to see them with the microscope. Now you have to see what type of media you need for the bacteria, specific bacteria, for transport, for growing. Okay. So types of cultured media. Culture means where you are going to culture the bacteria. That is the cultured media. Many organisms may be grown in the laboratory on artificial media because when they are not on the host, from where they are going to get their food, their all the things. So to check whether the bacteria is grown or not, what type of bacteria, you have to give them some environment where they can grow. That is the artificial media or cultured media. To grow a cultured microorganism, it is necessary to provide them proper nutrients and growth conditions, which they are getting for the host body. So when they are human, they are using the human as a host, they are getting all the nutrients from the host body, growth condition like the temperature, everything, environment, everything. So, but when they are not on the host body, where they will get that? But they need to, we have to see. Otherwise, we like to prevent them from growing, but to check what type of bacteria is there causing problem for the patient health. So you have to see which type of bacteria is there so that for the diagnosis or the treatment. So that's why you need to grow that bacteria. So that if you grow more in that, so you can easily identify what type of bacteria is there. That's why you need some proper nutrients and growth conditions. The growth of microorganisms on artificial material is referred to as the culture of microorganisms. You are culturing them, you are nurturing them outside their normal residing environment. And the mixture of nutrients on which the organisms is grown in the cultured medium. Isolation and definition of viable pathogenic organisms on culture media is standard for diagnosis of many infectious agents. So when you are developing them in the culture, then you are going to isolate it and then they can test it. What specific type of bacteria, what is their nature. If you then know what type of bacteria, then they can do the antibiotic specificity testing so that which antibiotic is going to be more effective antibiotic screening technique for the treatment of the patient or for the diagnosis purposes. So there are the choose appropriate culture media is essential for the isolation, growth, and final identification of the pathogenic bacteria organisms. Not all bacteria can grow in all environment. So the colonial characteristics of a particular bacterium, growth patterns, and biochemical reactions on the growth media are used to determine a presumptive identification. What is growth pattern? Whether they are diplococci, whether they are cocci, they are in cluster, they are in chain, they are in rod shaped, they are spiral, anything. That is the shape, their size. So it is going to give you an idea what type of bacteria is that. And there are some biochemical reactions you can check on the agar plate. So it's going to tell you what type of bacteria that you are going to may learn here to make specific identification what type of bacteria it is. So media used in microbiology are generally prepared from measured quantities of known substances that are formulated to give highly repeatable culture results. Nowadays, you can get the ready-made culture media from the companies. Because it comes sterile, you can use it under the biological safety cable. But sometimes or before or in the research laboratory, they have to make their own media. So if you have to make your own media, you have to know the composition of the specific media, how you are going to make it. So media of this type are produced synthetically and consist of the specific amino acids, sugar, salt, vitamins, and minerals needed to ensure the proper growth of certain bacterial species. So what specific amino acid there were, how much sugar will be there, what is how much salt is going to be there, vitamins or any other requirements, it is going to depend in bacteria and what's the dose or the amount. So there are different types of media used in microbiology. These are called the culture plates where you are going to, just give me a second, who is that? People. Hello? Hello? All these junk phones. Okay. So media, there are different types of media, solid media, liquid media. Solid media are used mainly on the plates or in the tubes. So basic media for the solid media, the basic platform is the agar. So agar is the basic platform for any solid media because agar, when you dissolve in the hot water and you cool it, it makes a solid base. But the soft solid bed is not like the hard wood. So prepared by adding agar to any nutrient liquid media. Agar is a sweet weed extract that is liquid when heated and solid when cooled. And it does not affect any bacterial growth. An excellent base or the platform for nutrient media. So you are going to dissolve everything in the media along with the agar, heat it, boil it, and then cool it. So in the agar medium, in the base, everything is there. So if you put the bacteria on the agar plate, bacteria can use all the resources needs to grow. So agar can be melted and poured into tubes or plates. As I told, it can put in the tubes or plates while it will solidify when cooled. Plates and agar slants are prepared with agar as the base. The same thing. In addition to agar plates or petri dishes, to observe growth characteristics of the microorganism, culture studies include use of agar slants for the maintenance of culture. Slants means slanting. In the tube is a slanting. You are going to show how it looks like in the lab for some biochemical studies or semi-solid media for motility. So this is the slant. This is the solid basic culture. This is the agar plate. It can be divided type. It can be three. It depends upon what type of agar media you are using. Generally, we use the sheep blood for in general. culture of the bacteria. And these are called the slant. So you can see the agar gel is not straight, it's in the slanting position. And it develops different colors as per the bacteria. And then we have the liquid media. What is the name of the liquid media? It's called broth. Broth, you know, chicken broth, beef broth, turkey broth, everything broth. That is the meat extracts from protein materials and intermediate product of protein digestion, that is the peptone or digested protein. Broths are used for all wound and anaerobic cultures. Who doesn't need the air? Anaerobic cultures. Broths are used for wound cultures and anaerobic cultures. They serve as a backup medium or enrichment broth if the suspected organism may be present in low numbers in the original specimen. So you did less collection or the specimen, in the specimen the number is less. So that time they use the broth instead of agar plate. The broth can be subcultured to agar plates for identification. So you primarily is low number, so you grow them in the broth, and once broth has developed some bacteria in the sufficient number, so then from there you can take that one in, you can inoculate in the agar plate to mix bacterial colony or growth. Okay, so broths are comes in like a either tube or in the conical flask. So these are that, and then you are going to put the bacteria here from the wound discharge or your sample or anywhere, then it's going to increase to 37 centigrade, and it needs to be very gentle shaking always. And then if you can see the bacteria is growing, in the clearness of the solution is going to go. You can see that it's getting opaque. You can see something is floating, moving. So that means the bacteria is growing. And it depends how much bacteria is growing, what is the amount is present in the patient. So types of media, there are 4 different types of media we used: enrichment media, supportive media, selective media, and differential media. So what is the enrichment media? It permits one bacterial pathogen to grow in presence of specific essential nutrients, only one type of, used to grow nutritionally exacting or fastidious bacteria. So enrichment media is good for fastidious bacteria. So what one example fastidious bacteria is L. pneumoniae, which causes the pneumonia. So buffered charcoal yeast extract agar supports to grow the L. pneumoniae pathogen that causes Legionnaires disease. I doesn't know what is the Legionnaires disease. What is that? Oh, kind of pneumonia. Severe form of pneumonia caused by inhaling this bacteria from water or soil. So it's pneumonia. Okay. And chocolate agar is called the chalk agar, promotes growth of fastidious bacteria. Supportive media that supports the growth of most non-fastidious bacteria. So these media do not give one organism any growth advantage over another. It grows whoever non-fastidious, it can grow all. If there is 5 different types of non-fastidious bacteria, they will grow everyone. But the selective this media, enrichment media, is going to allow only the growth of only one specific type of bacteria. So supportive media are the simple media that support the growth of most non-fastidious bacteria. These media do not give one organism on any growth advantage over another. They look neutral. The organism's metabolism affects the growth rate of the media. So how much the bacteria growth, it depends upon what is in the growth, nutrients or anything they are using from the media. Selective media. Selective media are prepared by adding dyes, antibiotics, or other chemical compounds that inhibit or suppress certain bacteria while allowing others to grow. Say for McConkey agar is called a selective media which contains crystal violet and bile salts to inhibit the growth of gram-positive bacteria. The another one is example is mannitol salt agar. It used to grow S. aureens. And Thyar Martin agar, this is very important, you have learned before also. It's used to recover or grow Neisseria gonorrhea. So you have to remember the names, which bacteria is for. So we have no different enrichment media, we have the supportive media, selective media, and then the differential media. Differential media is designed to recognize different bacteria based on the colony color. So you have a different types of bacteria, but they are going to take different color on their colony. So media contains nutrients, energy resources, and incorporate dyes or indicators. So bacteria that use them appear differently placed on the colored colonies. Each bacteria is not going to form the same color for the same dye. So they are going to use the same nutrients and all foods and environment, everything. But when they are starting growing, so they are using the color what is there, they are going to show different color. So that is called the differential media. Allows more growth than one microorganism, not like selective media of interest, but with morphologically different colonies. So if you watch the differential media, you can collect culture or you can grow different types of bacteria which may be present in the patient body and with an efficient rate. Example, McConkey agar, lactose fermenters are pink, non-lactose fermenters are pale or colorless. When you are growing these bacteria, what is lactose fermenters is going to appear as a pink. What that means? It doesn't have the peptidoglycan layer if you are using the gram-positive 10 only. But here you are using the differential media. So lactose fermentase is going to be pink. Non-lactose fermentase are going to be pale or colorless. So from there you can understand this colony is lactose bacteria. Mannitol salt sugar. Mannitol fermentation is called the who has the mannitol sugar on the structure. So they will turn out to yellow color. Blood agar. It various kinds of hemolysis. When the hemolysis occurs in the blood, they want to test you the blood agar to make sure which factor is making this problem. And they have three types of alpha, beta, and gamma hemolysis. So all media plates can fall into more than one category, that is MAC, selective and differential, CNA, selective and differential, or hectoin, selective and differential. So McConkey agar is a selective and differential media. And frequently for primary plating of specimen to isolate and differentiate gram-positive bacilli. Sorry, McConkey agar is a selective differential used frequently for primary plating of specimen to isolate and differentiate gram-negative. So they are doing the gram-negative bacteria. They cannot do that gram-positive. Gram-negative bacilli that ferment lactose will appear red or pink, and gram-negative bacilli that do not ferment lactose will appear clear or slightly pink. So light pink, or see the B, where there's a lightly pink in the agar plate, and this is who is a lactose fermenter. So they have the dark pink. So blood agar plate. Which type of blood is used? Sheep blood. Sheep blood is mixed with the agar to and other nutrients to make a sheep blood agar plate. Made from sheep's blood, all-purpose plate is mostly used everywhere. Most commonly used in primary plating that supports the growth of the most bacteria. Differential, useful to help differentiate hemolytic bacteria, especially Streptococcus species, based on the ability of the bacteria to lyse the erythrocytes, because it's called the hemolytic, because the bacteria lyse the erythrocytes of the blood cells in the agar. Hemolysis can occur alpha or green color, beta or clear, or gamma is red or no hemolysis. Okay. So how it looks like? When it's alpha hemolysis, you can see this one. When it's a beta hemolysis, you can see yellow things, what they are telling. Alpha, green. Beta is clear or complete. It's almost clear or complete like this. And gamma is there is no hemolysis. Whatever you put the bacteria is saying like this. So this is a bacteria. All are same bacteria, they have tested it. So it's alpha hemolytic, beta hemolytic, and gamma has no change. Chocolate agar is an enrichment medium that promotes the growth of fastidious bacteria prepared by adding blood to a nutrient-based medium. So just remember chocolate agar is an enrichment media. XB growth hemophilia. And you can just remember these two portions is too much complicated. That's why I told you what is chocolate color? Because the heat lies the RBC, causing the medium to turn brown. Sheep blood is more reddish because you are using directly the sheep blood. But here, they heat the RBC, lies the RBC and then heat. So it becomes turns to from red to chocolate color. That's why it's called the chocolate agar. Chalk. It's an enrichment media. It can grow a specific bacteria. So what bacteria is grown here? Hemophilus species, Neisseria species, which is called the gonorrhea, and meningitis. Meningitis, nosoria, and gonorrhea and meningitis, the Neisseria species, which can cause this. So there, the chocolate agar is an enrichment medium. They use the RBC, heat-lysed RBC in the medium. That is a chocolate. It is richer medium than the ordinary blood agar. And Then you can culture and you can say whether there's again what is the reaction has a positive, not what type of bacteria is there, Neisseria or Haemophilus. So this is the chocolate agar. You can see how it looks like dark. So this is the Neisseria gonorrhea. So you have streaked it here, and then you are going to learn the streaking techniques. And you can, these are called the bacterial colonies. You streak the main sample, and then you are again streak like this way. Then from here touching is going to this way, and ultimately it goes to this way. So here you can see the maximum colony, individual colony 1, 2, 3, 4 scattered. Here you can see one over another. Colistin nalidixic acid agar with blood is a selective and differential media used to recover gram-positive bacteria. That's why I hate this one. You doesn't need to remember all these things because it comes when it comes in the practice. You know which medium you have to use is already there. So okay, so this bacteria you have to use this. You doesn't have to know all this composition and other things, but you have to remember for your test. This is nothing I can help except you have to remember. What is this colcystin nalidixic acid agar with blood? It's in the blood, but they have some antibiotics. It's called the colistin and nalidixic acid. Inhibits more gram-negative organisms. The 5% sick blood aids in the differentiation of hemolytic organism, whether it's a alpha, beta, or gamma hemolysis. Then the hectoin enteric agar. This is also selective and differential media. The use for the culture of Salmonella and Shigella. And what are the indicators used here? Bromophenol blue and acid fuchsin. Salmonella, Shigella. So all the agar plates look the same, only the colors are different due to the presence of different things. This is called the McConkey agar, designed to selectively isolate gram-negative bacteria and differentiate them based on the ability to ferment lactose. The lactose fermenters is coming red or pink, and non-lactose ferment clear or colorless. In the same plate, it can give the difference if you have the lactose tolerant and intolerant. So there you can understand which bacteria is present. So selective acid inhibits the growth of gram-positive bacteria with crystal violet and bile salts in agar. How many media you can see? Phenyl ethyl alcohol agar is called the PEA, is also a selective medium. Most importance of the McConkey chocolate, sheep blood, what is another one? This doesn't come that often. McConkey is very common chocolate media, hectoenteric. Okay, so So, gross leakage, differential, selective media, differential media, types of media. So here the media start blood agar, chocolate agar, important. Cholecystine, this is doesn't come always, only you have to do the gram, recover the gram positive bacteria. Then the hecton enteric, this is mainly for salmonella and shigella, and they use the dibromophenol blue and acid fuchsin. McConkey agar is a selective and differential agar. You can use for the selective purpose and for the differential agar. So designed to differentiate them based on the ability to whatever I told you, fermentation of glucose, which fermented is red or pink, which doesn't ferment, it comes clear or cloudless. And it also inhibits the growth of gram-positive bacteria with crystal violet and bile salts in agar. This is phenyl PEA as selective medium is used to grow Staphylococcus and Streptococcus. This PEA is the active ingredients that inhibits growth of gram-negative bacteria by interfering their DNA synthesis. It can be made with by 5% C blood plus minus. Okay. This is without the blood and with 5% blood. Thier Martin, this is an important medium. Thier Martin agar is selective and enriched. Majority of the mediums are selective and enriched and used for isolation. We have already learned Neisseria, which causes the meningitis. It's a modification of chocolate agar. What we learned before in the chocolate agar, we can the Neisseria and the meningitis. It's a modification. It's more effective and containing hemoglobin and a supplement, which is now Isovital X, that provides NAD patients sorry, my voice is getting really difficult today. It's a modification of chocolate agar, what we learned, and it has a hemoglobin and supplement is called the Isovital X, so that it can help in the grow of bacteria, which is, forget about the NAD. This is more biochemical physiology. So just remember Thier Martin agar is a selective and enriched media and used for isolation of Neisseria, gonorrhea, and meningitis. That's all. You doesn't need to remember all those things. It's going to be too much. And I think there will be no question like that, but it's going to come that where you are going to grow. If they give the Neisseria, gonorrhea, and they give the options, Thermatin, chocolate agar, or any other kind of McConkey, where you are going to grow it. So Thermatin agar is a selective media and responsible for the growth of Neisseria. This is Sabord Dexor agar. This used to grow the fungi. So far we have learned is for the bacteria. For the fungi, we cannot use the medium which you are using for the bacteria. So what is the name of this one? This is sabourite dextrose agar. You can raise yeast with or with antibiotics. So it promotes the growth of fungi. It's a selective media. Because whenever you can specific, it's a selective media. It promotes the growth of fungi while inhibiting the bacterial growth from the specimen collected from the non-sterile sites. That's why you can raise with antibiotics. If you use the antibiotics, if there is any bacteria, they are not going to grow. So to inhibit bacteria, this sabordextrose agar has a low pH because fungus can go in the low pH and little bit low temperature also. So it should be incubated at 30 degrees centigrade. Chloramphenicol is included to inhibit the bacterial growth. That is one kind of antibiotic, and cyclohexamide may be included to inhibit non-pathogenic of saprophytic fungi. So cyclohexamide is block the DNA synthesis of the specific different non-pathogenic organisms and any saprophytic fungi. So this is SD or SAB, sabordextrose agar, is used to check the growth of fungi. So when some fungi, generally it should not be, but some fungi has some response or sensitivity to the antibiotics. So that times they told to use the SAB without antibiotics. Otherwise, in general, they fungi are developed or cultured with the antibiotics. These are called the colony of the bacteria, individual colony. It's a group of bacteria, and if there is a mixture of bacteria, different bacteria is going to make different colonies or the fungus. Here is the fungus. Chromogenic agar. What is the chromogenic agar? Chromogenic agar target organisms by their characteristic enzyme system that are able to metabolize added substance to release the chromogen or dye in the media. So some dye is already impregnated in the media. When you are putting or growing those bacteria or the pathogens, they will interact because they are developing, right? And they have used the dye or interacts with the dye because when they are using the nutrients from the medium and they interact and they cause the breakdown of the dye, which cause different colors or no colors. So it's called the chromogen is then detected by direct observation of a distinct color change in the medium. Its advantage fast results compared to traditional methods, reliable visual detection. Sometimes no further testing is required. Additional testing is possible directly from the media. So see, there are some dye is impregnated already in that. The E. coli is there, they are going to immediately turn the color to blue, or if there is a pneumonia is going to change the color to pink. So different colors can be developed and it can be easily visible. That's why day by day new methods are developing for helping more efficient and fasting results. So Salmonella Shigella agar, this is specific from the name you can understand. This agar is specifically to culture Salmonella Shigella. Originally developed as a selective medium for the isolation of Salmonella and Shigella, it was also developed to aid in the differentiation of lactose and non-lactose fermenters from the clinical specimen, which is from the suspected food and others samples to make sure whether you have the infection with a bacteria, the Salmonella, which is lactose tolerant or intolerance you became due to presence of this pathogen. The basis of differentiation depends on the fermentation is same. Okay. This is the Salmonella Shigella, how it comes. That's why I hate this one. Is so many medium. It's going to be really difficult. So let us see eosin methylene blue agar, EMB agar. This is the selective stain for gram-negative bacteria. It contains dyes that are toxic to gram-positive bacteria. So if there is a gram-positive bacteria, they are going to kill the gram-positive bacteria. Only the gram-negative bacteria is going to grow there. It's called the methylene, eosin, methylene, blue agar, EMB agar. It blends two tenths eosin and methylene blue. From the name you can understand in ratio of 6:1. Doesn't need to remember. A common application of this stain is in the preparation of EMB agar, a differential microbiological media which slightly inhibits the growth of gram-positive bacteria and provides a color indicators distinguish between organisms that ferment lactose and those does not. When it comes to different color, it's the metallic, it's the lactose positive. Otherwise, it's grow like this. But when it's lactose positive, the bacteria, when they are placed on the agar, it looks like this. Change the color. Just give me a second. I'm coming back to the washroom, okay? And then probably we'll go for a little bit break. Okay, so just give me a second. Oh, sorry. My microphone is off. Woohoo, sorry. So I just teach you guys this one. So salmonella, this one has been done. Eosin has been done. So this one is for the Campylobacteria. So this is mainly used for the fecal samples. And there they use some kind of antimicrobial things in the medium so that the normal fecal flora, which is always present, they are not going to grow. So inhibit the growth of the normal fecal flora. If there is any other bacteria, Campylobacter, so then that can grow. So this is the, well, it's the Campylobacter. So if there is anything, it's going to grow. But the E. coli is the normal flora in the fecal sample. So they put some antimicrobial agent so that there will be no growth of yeast and also not normal fecal flora. Only if there is an infection with Campylobacter, those are going to grow to make sure there is an infection or not. So they are using cefoperazone to inhibit the normal, gram positive and gram negative flora, and vancomycin to gram positive, any kind of gram positive microorganisms, and amphotericin for the inhibition of the yeast. So this is up to this of the media. Now it's a broth and slant media. We are going to learn next up. Okay. Okay. Let's to go for break.
Yes, please, miss.
I know it's so boring chapter. I don't like this microbiology. It's so boring. From this part is boring. Yeah. So from this part is boring. Till before that it was okay. From this part I don't like it either. It's just a remembrance memory and you doesn't need it. Only you have to give the exam. That's why. Otherwise everything is there. Is you know which one, then you are going to use the same plate or something like that. It's too much information. But
Yes, miss.
yeah, what I can do? They put in the course. So I have to tell you, but I'm trying to tell you the short. What is the name of the media and is used for what purpose? If there is special anything addition, just remember that. You just don't need the whole slide. Okay?
Okay, thank you,
Okay, okay,
miss.
okay.
I'm also.
So what time are we back, miss? 30 minutes? Okay, I guess so.
What what happened?
Yeah, miss.
What time you want to come back?
11:30, ma'am.
That is going to be too much.
11:30.
11:30 is going to be too much.
11.
Okay.
11:20.
Okay, okay,
11.
go for it. We'll do today and tomorrow you are going to take the exam in the first half, right? Thursday? Tomorrow is a Thursday, right? Tomorrow you guys are taking the exam.
The exam is on
Mom,
Friday.
Friday.
I thought it's Friday, miss.
No?
Yes,
No?
Mom,
It's
Friday.
on Friday, miss.
We have the schedule in the
Really?
email. Yes,
Yeah.
Mom,
Exams
you
are
send.
Friday? Okay, so then we are going to complete everything tomorrow. Then you will get some time to some review, and then you can do the exam on Friday, and the rest of the things you can review on the weekend. Okay?
Okay,
Okay,
Mom. Thank
Oh,
thank
you.
you,
I
miss.
have to send an email remind Vera for the Tuesday, right? Exam is going to
Yes,
be the
Mom.
Tuesday. Okay, Tuesday
Yes.
computer room. What is the assignment?
Yeah.
Yes, test 2 doesn't have too many questions, only 40, but test 3 has a lot of questions. It's the final one. Yeah. Okay, so come back at 30. Okay? Okay.
Okay, thank you.
Okay, not
Thank you,
everybody,
ma'am.
Mom,
no,
thank
don't
you.
leave the team. You go for break, but don't leave the team, okay?
Yes, ma'am.
Yes.
Yeah.
Okay.
What?
Ma'am, your voice is not coming.
I know.
Oh,
I'm looking something to take in my brain.
okay.
Thank you. So what is bird? Okay, come back to again the pudding things. So we have so far learned about the agar, that's the solid media. Now we are going to learn little bit as much as possible with the short time. The broth, that is the liquid media. Liquid media is another name of liquid media is broth. There are different types of broths, like the different types of agar plates, depending upon what is going need to be grown, which bacteria or which fungi, which is going to be need to be grown. So selenite broth. It is also an enriched media. Enriched means where everything is there to encourage the growth of bacteria. So why the broth is important? Because in the broth, even a bacteria is very small in number, if you put in the broth, it can grow. So even the small number of specimen or pathogen is present, it can be detected, like the salmonella feces, which is from the foods or environmental samples, to make sure, to check whether there is any salmonella is present in the food or in the environmental samples, not actually the patient, to make sure whether there is nothing in that things. So suspicious colonies from the culture plate are tested directly for the presence of Salmonella somatic antigens by slide agglutination. These are too much things. Don't need that one. So mainly light broth is important to use food detect small numbers of Salmonella feces or to check is there any contamination in the food or in the environmental samples. Sometimes for the quick assessment from the culture plates, when you grow in the culture, this fecal parasite, fecal bacteria or fecal pathogen, they can be taken directly on the slide and tested from the agar plate or the culture plate. So that's why a presumptive diagnosis of salmonellosis can often be made within 24 hours of receipt of a specimen, although confirmation may take another day. Generally, the blood culture report, any report, comes within 48 hours, so 2 days and 2 nights. Before that, you cannot get the answer. So that's why, but in this case, they can do it within 24 hours in the agar plate. So selenite broth is made up of disodium hydrogen phosphate. It's useless to remember. Coloniform bacilli only is important that it doesn't allow to form, which is coliform bacilli. Only they allow the bacilli which cause the dysentery. That is the Salmonella typhi and Shigella sonei and Shigella flexneri. So this selenite broth inhibits the bacteria which is coliform type. But who forms the what is coliform? Which cause the colitis. That is the coliform bacteria. But it helps to grow or culture the bacteria which cause the dysentery. Which cause the dysentery? Salmonella typhi, Shigella sonei, and Shigella flexineri, so Sigalapicis. There is triple sugar iron agar slant. Triple agar sugar is called the TSI slant. It contains glucose, sucrose, lactose, phenol rate as a pH indicator, peptone as nutrient source, sodium thiosulfate, sulfur source, and ferric ammonium citrate. So these are the all composition for this slant. Proper inoculation of the media includes stabbing, use a straight inoculating needle, the butt through the center to the bottom, then sticking the slant. They're going to show you how to incubate, how to inoculate. It's called the inoculation of the bacteria in the plate or in the slant culture. So the TSI medium, it's the identification of gram-negative bacilli, is used to test ability of gram-negative bacilli to ferment glucose, sucrose, or lactose and produce hydrogen sulfide. Recently tested as denoted by the black color to test the ability of bromoactive bacilli to ferment glucose, sucrose, lactose. Fermentation of sugar is accompanied by acid production, which is indicated by a change in the color of the phenol red. So basically, in the plant, there are all the nutrients at the same times. There are some dye is impregnated. When it comes in contact with the bacteria, and depending on the nature of bacteria, it started multiplying and develop the color. So it says the fermentation of sugars is accompanied by acid production. If there is a sugar present, what is going to produce acid, and which acid is going to change the color of the phenol which is present inside the dye to red from yellow. So ferric ammonium citrate and sodium thiosulfate are present for detection of hydrogen sulfide as noted by a black color. And bubbles or splitting of the agar indicates gas production. That means there is gas. Producing bacteria are there. So this is basically is called the slant culture. So you're going to put the bacteria through the center, go down, and then you are going to strike it. Then you can see different coloration. If there is anything that is the is telling that something has dropped. Okay, so you are not going to use another except the non-sterile one, long one. You are not going to use anything because you are going to take out that one. But otherwise, you doesn't need to take it out until the exam is done, test is done. So TSI test for fermentation of glucose, but lactose and sucrose, H2S production, gas production bacteria. A acid slant acid with gas. You can see the cracks. So that means it's producing the gas. It states yellow, no change, only the gas production is fermentation of glucose. Alkaline slant acid, but with black precipitate. This is C, it's called the alkaline. See, this is also alkaline slant, alkaline bat, but it changes from red and no fermentation of 3 sugars. There is no fermentation of the sugars. When there's a black precipitate, glucose is fermented because it's produced the H2S. Here, there is no fermentation, so there is no change, basically. C is alkaline slant, red is no fermentation of any of these sugars, and D uninculated tube. So that means there is no bacteria or nothing has been inserted there, inoculated there or put there. So it's nice, organized and everything. When you are putting something which is called there is no fermentation of the sugar things, so you can understand wherever the sugar forming bacteria or anything is there is nothing, you're bubbling or sucrosing, you are not from the, your body is generating gas. So urea agar. Urea agar is used to test the ability of a microorganism which use urea for their growth. So it produces conversion of urea into carbon dioxide and ammonia. So you can get the smell of ammonia. So you can understand these microorganisms is producing urea, producing ammonia from the medium where there is carbon dioxide and ammonia. They are going to break down the urea to carbon dioxide and ammonia. The presence of ammonia raises the pH of the medium as indicated by a color change of the phenol red indicator to a bright pink red or magenta color. So it's already there. So when there is a pH is increased, so what is going to be change of color will be there. As we know, there is a change in color is possible to leak. Okay, so the organism is straight on the slant. The butt is not stabbed. It's just go straight into the slant. It's not stabbed. Some organisms produce small amounts of ammonia, resulting in a pink colony all in the slant. In others, both the butt and the slant will be pink. So this is the urease test. It's called the urease test as it is using the urease enzyme to break down the urea. So it said the test organism is cultured in a medium which contains urea and the indicator phenol red. So this is the negative. When the test is negative, the slant is going to be like this. When the test is positive, that means there is urea producing, ammonia producing bacteria is there. So it's going to come like the positive. So that means when the strain is urease producing, the enzyme will break down the urea to give ammonia and carbon dioxide. So that way the ammonia is going to rise in your body, even ultimately in the blood level. So with the release of ammonia, the medium becomes alkaline as shown by the change of the color of the indicator pinkish red. And this is negative, that is no uninoculated, and this is the reaction of the bacteria is become pink in the slant culture. Let me see one thing, how much we left for tomorrow. That's one college. Just give me a second. Looks like too much information in one day. LA. Mm, this one, 5. Six. Newsletter. So now we are going to prepare the media. Bacteria and fungi can grow on or in microbiological media for various types, different types of media. The medium that is used to culture the microorganism depends upon the microorganism. As we learn, different media, different slant, different nutrients, different dyes needed for their culture. And different nutrients may be added to the medium, making it higher in protein or sugar. Various pH indicators are often added to the differentiation microbes so that we can change the color by the change of the pH or some by biochemical reactions. Other added ingredients may also be growth factors like sodium chloride, pH buffers, which keep the medium from straying too far from neutral as the microbes metabolize. The old way to make the media was by the cookbook method, adding every ingredient bit by bit. The only time that is done today is when making a special medium to grow a certain funky organism, where particular growth factors, nutrients, vitamins, and so on have to be added in the certain amounts. So this is you have to make by your hand totally. Otherwise, nowadays the medium can be purchased. So this medium is called chemically defined medium or synthetic medium, which you have to make it as per your supervisor's guidance or whatever is needed. If it is needed, it is going to be special type of medium. Fortunately, the most common bacteria that we want to grow will do nicely with media that we commonly use in the lab. Some of the media is bought, commercially prepared, and some is produced in the prep area in the lab. Some certain types of medium have unknown ingredients or sometimes unknown qualities. That is called complex media. So it is really very simple to make complex media these days. Rehydrate the powder from the medium, stir and boil the agar medium to get the agar powder dissolved. First, you have to dissolve the agar powder in the water, then distribute the medium into the tubes, autoclave to sterilize the tube media, particularly the agar medium, because everything dehydrate the powder from the medium. So you have the medium in the form of a powder. You are going to wait, then you are going to rehydrate, means add water. Ultimately, how much, what is the proportion? So when from the what is called the powder you have, it can come as a powder in a bottle. And they knew how to make the proper solution now. Same the, it's a 2%, 5%, 6%, at this one, at that one, you know the math now. So as per the SOP, you can make the medium. Generally, you don't have to make it, but if needed. So examples of making media: objective, how to make media, how to sterilize it, and how to distribute it in different formats. So you need one spatula, 30 medium, non-sterile test tubes, 2 plastic waveforts, 1 test tube rack, 1 jar, nutrient, triptychs soy, brother, yellow caps, jar agar powder. So this is the ingredients you need, and also you need these things: 15 green caps, 1 2 liter inland flask. You know what is Erlenmeyer flask is. So we are going to take 1 liter that flask, 1 pipette pump. You know how to increase the volume and decrease the volume. It's called the pipette head. So pipette pump to up and down the solution, magnetic star bar so that you can swirl the medium so that it can get well mixed. Pipette disposal jar where you're going to dispose of the pipette, a graduated cylinder to measure, several non-sterile glass 10 ml pipette so that you can divide it, transfer it one by one. So the procedure, see the diagram below, takes for the entire production. Marks making the TSB, that is the broth, by pouring 250 ml of distilled water into a 500 ml of flask. What is the percentage? It didn't say. So pull the star bar and turn on the star plate so that the surface is now just separate. Add 3.25 grams of TSB powder to this flask and allow it to dissolve. It happen quickly, but no heat is need to be applied at this stage. There is no need of heat. Okay. So once powder is dissolved, pipette out 10 ml aliquots into 15 tubes. That's why I take 15 tubes and 15 caps. So we are going to take 15 ml of tubes in each that slant tube, and then you are going to cover it with a green cap tube. So with the remaining solution, still stirring at 2 grams of agar powder. Next step, you will require to apply heat to mixture. Before you do this, however, you should be aware the agar has a strong tendency to boil over when it reaches 100 degrees centigrade. Base temperature is 60 degree. So this is the diagram. Each table makes 250 ml of medium, 250 of distilled water in a flask with stirrer. So 15 to 10, 15 10 ml TSP tubes are removed. You add agar powder, then boil, and then you are going to make 15 5 ml TSA tubes. So notes while making the media, someone in your group should be watching the flask at all the times. Once the steam is coming up, because it boils very easily. At the first sign, the mix is near boiling. Remove it from the hot plate with the paper towels around the flask neck. Do not simply turn up the heat, letting the flask sit there. The metal plate retains a significant amount of heat, and turning up the heat will not prevent the flask from boiling over. Folded paper towels allow you to grasp the flask neck tightly, yet not burn your hand. So then turn on the heat to setting 9, not high. Make sure that the magnetic bar is stirring the solution. When mixture boils and the agar dissolves, it will turn clear, deeper tan. Remove it from the heat and pipette 5 ml aliquots into 15 tubes for slants. They will not be slant until you remove from the autoclave and tilt it to the side to solidify. So after that, you have to do the autoclave in the steam sterilization, and you have to put it in the slanting condition. So they are going to make a slant. Cover the slant tubes with yellow caps. Leftover agar medium will be poured into flux for the class to be autoclaved. From this point on, yellow caps will be used for the nutrient agar slants. Also note, if the agar solidifies in the tip of the pipette, dispose of the pipette in the pipette jar and get another one. So prevent this from happening, either pipette out all the tubes at the same time or leave the pipette in the flask on the melted sugar. If you just take it out, it's going to block the pipette because it's getting cold temperature outside, and you are putting the flask on the hot plate so that it's not going to not you just turned up the heat, but it's still melted. Sterilization and autoclave. When microbiological media has been made, it still must be sterilized because of microbial contamination from your air, glassware, hands, environment, that is the air. So within a few hours, there will be thousands of bacteria reproducing in the media. So it must be sterilized quickly. As soon as you make, you should be ready for sterilization before the microbes start using the nutrients up. The sterilization process is 100% kill and guaranteed that the medium will stay sterilized unless exposed to the contaminants by less than adequate aseptic technique used to expose to air. The prepared media is distributed in different ways depending on from one form is making. Both agar and broths are dispensed into the tubes and then sterilized. Agar slant tubes are sterilized and then the rack is tilted to allow the agar to solidify in a slanted fashion. So for making the slant, this agar tubes is going to be sterilized first. Then they are going to put in the slanted rack so that it became from a slant that in the tube. Agar medium to be poured into place in sterilized in a flask and then poured afterwards. So sometimes you can sterilize the total agar medium and then under the biological safety cabinet in the sterilized condition, you can put the sterilized medium. So not all media or solution can be sterilized by an autoclave because it's a certain high protein solution such as urea, vaccines, serum will denature the extreme heat, and so they have to be filtered, sterilized without heat. What we learned, right? The antibiotics, vaccines, some solution. You have to do the filtration. What is we call the millipore filtration, so that it can all the bacteria can be anything can be filtered and the solution is going to be sterilized. The solid media, the liquid nutrients are mixed with the melted agar, heated to around 100 centigrade and poured into a petri dish or agar plate. For the liquid, you need the agar as a base or platform. Plates are left to solidify. It takes some time because it's melted agar, right? When you are going to put it in the plate, it's going to take some time to solidify. Once the medium has been cooled and solidified, the petri dish is inverted so that the lid is on the bottom. not the lid is on the top. This is the question. It comes several times. After sterilization, how are going to put the, or after making the agar plating, how we are going to put the agar plates. The tops or the cover should be at the bottom, and the agar plate, which is solidified agar plate, is going to be sit on the top of the cover. Why we do that? Because from the agar plate there may be some condensation. If the condensation, it should go in the lid, which is down. So when you are taking, it's not going to, if you put the cover on the top, the condensation is going to get back to the agar. So that you have to put it upside down. So that any condensation which is coming on the cover plate, it will stay there in the down. It's not going to go in the reverse. It's not going to go up to contaminate or make the agar plate not suitable for inoculation. So plates are also stored in an inverted position after inoculation. Even after you put the bacteria there, you have to put them in the same position. So they are labeled on the bottom of the portion of the plate containing the medium. Slant cultures are solid media in a tube and can be used for storage, transportation, or biochemical testing. When preparing slants, the medium is dispensed in the tubes and then autoclave and then make the slant. So liquid media overview. They are prepared by mixing nutrients to distill water and placing broth into the test tube. The liquid media. The broth tubes are then autoclaved or heated 100 degrees centigrade in the autoclave for sterilization. It's not only the 100 degrees. The media is then cooled to room temperature before inoculation. Inoculation means insertion of the bacteria. Wound and anaerobic cultures are processed using solid and liquid media. So anaerobic and any wound can be done by both aerobic and non-aerobic, anaerobic cultures. When the microbiologists suspect that the pathogenic organism may be present in the low numbers in the specimen, a broth media can be used to recover organism of insufficient number that we already discussed. So when bacteria grow in broth medium, the appearance of the middle will be changed from clear to turbid. I already told you, because first the medium color of the broth is going to be very clear. There will be color. Sometimes depending upon the what type of media you are making, with dye, without dye, but it should be clear. But when the bacteria will grow, start growing, there will be presence of lot of bacteria. So microorganism. Ultimately, the solution is instead of clear, is going to start turbid, not clear. So that is the sign that you may have infection, whether it is a UTI or anything, but you may have an infection. Blood cultures are another example of using liquid media. Blood is collected, placed in your broth medium in bottles. When you are collected it, incubated, and if positive, can be gram states and subcultured for identification. The following media chart can also be found in D12 unit folder 5. You will see there is an enormous variety of media available. So this is the chart of different media, what type of media it is, what is enriched media, what is selective media, then what is the use of the media, what type of enrichment they are giving, and then different types of selective colors or agents, and what is the differential agent. So this is the chart for your guys' knowledge. What is which media, what type of media, why use, what type of things, supportive things are there for the detection of the disease, the bacteria. So how you are going to do the quality control? Always you have to do quality control. Store media based on expiry dates, newest in the back, oldest up on the front, as usual. Do not use any expired media. Store all media based on the recommendations from manufacturer. Whether it is a room temperature, whether it is a refrigerator, whenever minus temperature, it depends upon the so store all media based on the recommendations from the manufacturer. That's it all. When opening up media to plate, look for any growth. When you are going to use the plate or the media, first you have to check whether there is already any growth. There may be chance during the process from making to sterilization and store. It may contaminate anytime, anywhere. So every time you have to check thoroughly everywhere so that there is no growth. So when using the broth, look for any turbidity in the media that would demonstrate contamination growth before inoculation. Commercially prepared media have QC performed on them before leaving manufacturers by batch and lots. So they do their own QC and then sell it. So Clinical Laboratory Standards CLSI has developed recommendation for the use of QC testing. If manufacturer has followed the CLSI recommendations and performance media is consistent, then additional QC testing is not needed in the clinical lab. If they give you all the information that has been tested by CLS followed guidelines, you doesn't have to do the QC, but always better to QC because you never know what is happening. So media quality control, all media prepared in the lab must be tested before being used for routine culture. Use known QC strains and plate on the media that you demonstrate the growth to make sure your plates or your media is working. Should grow as expected with no mixed cultures. QC organism, they send you some couple of virus or bacteria mainly. So then you are going to use that one to inoculate or in the agar plate or in the liquid media to grow, to make sure everything in your system is working. So QC organisms specific strains that demonstrate the specific result. So if it is a bacillus, it's going to be the bacillus. These are purchased by the lab and used to test the media. And lab maintains a stock of QC organism for QC testing. Always you have to keep the logbook. And this media can be stored in minus 80 degrees centigrade. So media storage generally stored under refrigeration 4 degrees centigrade to prevent deterioration and dehydration. If media requires special storage, then information will be provided with the media when it's going to come from the supplier. So you have to keep, these are all the agar plates. Different types of agar plates, sea blood, choco blood, everything. And these are going to be put upside, lid upside down and in the microscope. So if the media requires special storage, this information will be provided with the media. Keep media in plastic packaging until being used. That means it is going to be inside a plastic packaging until it is used. Because as much is used, if you doesn't work it within some time frame, the gel becomes dry. So you are keeping the media in the plastic packaging until being used. But when you have to use it, what you are going to do? You are going to take out from the refrigerator and start working? No. For any biological samples, what is the rule you have to follow? You have to take it out, bring them at room temperature before inoculation, as bacteria can be sensitive to temperature. If you give inoculate on this bacteria in the cold condition, the bacteria will feel cold and then probably doesn't go. So you have to bring at room temperature. Labs keep good tabs on what their media inventory and this have a minimum level that triggers reordering of the stock. That is the inventory you always have to do. When media inventory arrives, the received stock is checked against the purchase order to confirm everything has arrived. So in the media is not the one powder. Maybe as a combination is full powder, maybe different components. That's why they're telling when you receive the things, see whether everything from the previous order or previous, what is called the leaflet that you have received everything. So when stocking the new media in the fridge, it is important to rotate the old media in the front, like the inventory. So the oldest expiry date will be used first. Put the new stock behind the old stock. You guys know when getting media to restock, you set a bench, look at the expiry dates. When removing, not everyone rotates the media properly. Yeah, they doesn't move the old one to front and so precautions, storage, shelf life, quality control. So everything has been written in the medium storage example on your humus sheet. What is a complex medium? Hmm? What do you mean by complex medium?
The composition is more than one, ma'am.
Yeah, different types of things has been included. Let us see the answer. Many different chemicals, some synthetics are added to the medium. Why pH vapors added to growth media for microbes? So microbes metabolize, right? And they change the pH. So that's
Yes,
why the
ma'am.
pH vapor are used so that they can keep the pH balance. Why do you have boil agar solution before dispensing in the tubes?
Kill any microorganism that has been there,
No,
make
no,
it
no, no, no,
sterile.
no, no. Yeah, yeah, no. Otherwise, you cannot put it.
Sterile,
You,
yes,
you
ma'am.
need to dissolve the
Oh, before.
ingredients.
Okay,
Dissolve
ma'am.
the ingredients because agar it needs to be boiled at the heat.
Yeah.
Yeah, at home temperature, agar solidify 30 to 40 degrees centigrade. So always better to the temperature at 60 degree so that you can pour, you can do whatever making the media. If it is more than 100 degree, close to 100 degrees, starts boiling. So we always put the hot plate temperature and now 60 degree. So this is, differential and selective. This is type of questions is going to come. This is some examples. What type of media is McConkey agar plate? Differential and selective. Review question is the agar plates that can demonstrate the difference between the lactose and non-lactose fermenting bacteria. Indicate which is the most commonly used place in the lab. So this you have to do by yourself, okay? So you guys can read what is the meaning of the agar place difference between the lactose. We just go through that one before. Lactose and non-lactose fermenting bacteria and
Yeah.
which is the most commonly plates used in the lab. In the that is the sheep blood agar. Okay, so just
Yeah,
I
the
give you
blood
the
bank.
one answer. Yeah, so you can
Yes,
get
ma'am.
work on that. So I think it's going to be it's already too much information for the day. I agree with you guys, but what I can do, this is the we did the unit 5. Then we can go to the unit 6 as much as possible. Okay. And I will try to give you some time if you can just complete the unit 6. And then I'll try to give you some time. Today's Thursday, right? Thursday. So you can review a couple of questions. If I can give you, or if you guys can stay a little bit, we'll see how long, when I can complete. And then you guys can do something. And the Friday after your class, maybe we are going to complete the rest. Or not after your exam. Is that going to be okay for you guys? We'll see how much you can cover tomorrow.
Yes, ma'am.
Okay.
Yes.
So if I can, I'll give you probably 1 hour or 1 hour and a half time. So by this time, you guys, you have some time now. If you guys want, you can work on your, what is called?
Yeah.
Yeah. So yeah, and then probably you can get some time to review questions. And then you can appear on the exam on the Friday morning. And then after the exam, we are going to finish the rest of the topics. And you guys are going to be ready for your exam. Okay?
Yes, miss.
Okay, I know it's too much. I told you, this is too much to remember. Not only
Yes.
understanding, that is the thing, not only memorize.
Miss,
Yes.
can I go now, please? I have a 1 o'clock appointment.
Yeah, go.
Okay, thank you.
Go. Yeah, you can take care of these things. Yeah, yeah.
Yeah, thanks a lot.
Okay, no problem.
Okay,
I know it's
bye-bye.
going to be a little bit challenging of this. Unit 1 to 3, first test exam is not going to be that bad, but test 2 and test 3, you have to, but you have time for that, to do the thorough study. If you doesn't use the time, the long weekend, going somewhere, and this you can do, but you have to do the study. I doesn't like microbiology, especially the medium and this type of portion. Other things is okay. Staining, different type of staining, why is staining, how we can did that microscope and other things. But this medium and this and that, because there is SOPs, right? If you have to make some SOP, there is the lab, there is the SOP. You are going to read it and do it. Anyway, enjoy the medium.
Thank you, miss.
Okay.
Yes.
Okay, so
Thank you, miss.
yeah, okay. I'm going to stop sharing now. I have to do the attendance. I have to send email to Vera, couple of things. Okay? I will be
Okay,
on
I
mic.
got.
Okay,
Okay, ma'am. Thank
Take
you.
care.
okay. Yeah, now is study period. You are not leaving. She has an appointment, that's why she is going. Nobody is going to leave. Do your study. Do your study.
Okay, ma'am. Thank
Yeah,
you.
yeah. Okay. I know. I told you guys, microbiology is a difficult one. Remember, it's not that difficult. I should just not difficult. It's very hard to remember. It's so close and so many types. Which is what? Basically, remember mostly which medium is used for which type of bacteria. That is more. And what is the basic ingredients? Is there any indicator? And for the stain, which stain is for which specific bacteria? And what are the stain, primary, secondary, or counter stains? Okay, so good. Okay, let me send an email to Vera now first. So Vera. Stop. Unit 5 is done. I don't think there is anything indicating the unit 5 after this one. Shall slip to unit 5. Oh, sorry, read up to units. Which one I did? 7. Unit 5. Oh, so this is we did up to unit 5. There was nothing after that. I don't know. Oh, this is the type of chart. This they gave it to you. Okay. Yeah. Sorry, there is one video I forgot to show you.
In order to grow, cells need very specific environmental conditions: food, energy, proper temperature, and humidity. When growing cells in the lab, we have to create these conditions using culture media. Solid culture media is a mixture of agar and nutrients poured into petri dishes. In this exercise, we'll demonstrate the steps for preparing brain heart infusion agar. Always check the lot number and expiration date. The right panel lists the composition of the media and the final pH. First, add 100 milliliters of deionized water to a graduated cylinder. Place a stir bar in a 250 milliliter flask. For the next step, we need an analytical balance. Place a weigh boat onto the balance pan. Press the tare key to zero out the weigh boat. Use a lab scoop to add 5.2 grams of brain heart infusion agar to the weigh boat. Now add the powder to the flask. Add about 75 milliliters of deionized water. Place the flask onto a stir plate. If you're using a combination hot plate and stir plate, be sure that the heat is turned off. After about 2 minutes, add the remaining 25 milliliters of water from the graduated cylinder. Stir the solution until all of the visible clumps have been broken down. Cover the flask with aluminum foil. We're using foil cupcake wrappers because they're the perfect size. Place a piece of autoclave tape over the foil and label it with the media name. Next, use the autoclave to sterilize the media. The heat from the autoclave will also help the agar to properly dissolve in the water. Power on the autoclave and make sure the drain valve is closed. Add deionized water to the level indicator line. Place the flask of culture media into the basket. Insert the basket, close the lid, and turn the handle to create an airtight seal. Use the control panel to set the mode to sterilize, the temperature to 121 degrees Celsius. We'll run this cycle for 17 minutes. While it's running, enter the date and time and the operation details in the log and initial. Once the cycle ends and the pressure gauge reads 0 psi, use heat-resistant gloves to slowly open the lid and remove the sterilized flask. Back at the bench, place the flask onto the stir plate and stir the media very gently and allow it to cool to about 45 degrees Celsius. Pour the media into the petri dishes. The amounts don't have to be exact, but be sure to cover the entire bottom surface, which is about 20 milliliters for standard size petri dishes. Once the
Here we
media
put
has
the
cooled, it will
piping.
solidify and turn opaque. Here you can see the difference. The petri dish on the left has set up properly. Turn the plates upside down to prevent moisture from condensing on the agar surface and store them in a plastic bag until you're ready to use them.
There is nothing there. This is the video. Close. Oh, this guy's. Oh, this is yours. Okay, so then it's divided.
Armando Hasuringan, Biology and Medicine videos. Please make sure to subscribe, join the forum and group for the latest videos. Please visit Facebook, Armando Hasuringan. In this video, we're going to talk about the mode of action of antibiotics. So we're not going to look into extreme detail, but we'll just look at the overview in a way. So mode of action of antibiotics. Now we have to know that antibiotics either destroy or slow down the growth of bacteria. So therefore, if it destroys a bacteria, it is bactericidal. If it slows down the growth of bacteria, it's bacteriostatic. Here I am drawing a typical bacteria with cell membrane and cell wall. The cell wall and cell membrane structure I am drawing is characteristic of a gram-positive bacteria, but we will just assume it is any type of bacteria. Within the cytoplasm, within the bacteria, we have a circular DNA. We can have RNA being synthesized and ribosomes here. Ribosomes will synthesize proteins. The first type of antibiotic are the ones that I want to talk about are the ones that target the cell membrane. These antibiotics will disrupt the cell membrane function, which means that they disrupt the phospholipid bilayer. So here is the cell membrane and here is the cell wall, known as a peptidoglycan. They are different. The antibiotic binds here to the cell membrane. It will alter the cell membrane structure and will make it more permeable. This will just disrupt the osmotic balance, causing leakage of cellular molecules, and essentially it will increase the water uptake, leading to cell death. Examples of these drugs are polymyxins, which I spelt wrong, and polines. Now, polines are actually specifically used against fungal pathogens, but that's just a note to know that you can use polines that target cell membranes in fungus. So polymyxins and polines target cell membranes. Other antibiotic classes target the cell wall. These antibiotics will inhibit cell wall synthesis, so these drugs are therefore bactericidal. They will cause bacteria to die. Example of these drugs are penicillin, cephalosporin, and bacitracin. I hope I pronounced that right. All these drugs have different mechanisms of action when it comes to inhibiting the cell wall to synthesis. A good thing about these drugs is that it won't affect human cells because human cells do not have peptidoglycans. We do not have cell walls. Penicillin and cephalosporin are beta-lactams. And then you have the bacitracin and other glycopeptides that prevent the synthesis of cell walls as well. Antibiotics that disrupt the cell wall is pretty extensive, and so hopefully I will make a video on that. Cell walls are important to bacteria. Disrupting the cell wall or preventing in cell wall synthesis will result in cell death. The next type of antibiotic class is the ones that can inhibit here DNA synthesis, or what's occurring here, RNA synthesis. So these antibiotics will inhibit RNA and DNA synthesis. So here the bacteria are typically having DNA replicated, of course, like so. So we have antibiotics that prevent from DNA from being synthesized. These are quinolines and also a class known as nalidixic acids. And then, of course, RNA can be made from the DNA. And there are drugs such as rifamycin, which prevents RNA synthesis and therefore protein synthesis. Other classes of antibiotics also inhibit protein synthesis itself. Protein synthesis is carried out typically by ribosomes, which translate mRNA into proteins. Bacteria need to make proteins in order to survive. However, there are antibiotics that target either the 30S subunit or the 50S subunit of the ribosomes. Erythromycin and chloramphenicol target the 50S subunit of ribosomes, whereas there are tetracycline, streptomycin, and gentamicin that target the 30S subunit. Either way, this will disrupt the ribosomes from making proteins, which will result in the bacteria obviously unable to make proteins, so the bacteria is pretty much useless. Thus, the bacteria can still be alive, but it just is unable to make anything. And so these antibiotics that target protein synthesis are bacteriostatic. They prevent bacterial growth. The last type of antibiotic are the ones that inhibit folic acid metabolism. So normally, this green thing here called PABA, which stands for para-aminobenzoic acid, it's a precursor to folate. So the bacteria can metabolize PABA into folate. There are antibiotics out there that inhibit folate or folic acid metabolism. See, PABA normally can be metabolized to folic acid within the bacteria, and folic acid or folate is essential for the synthesis of adenine and thymine, two of the 4 nucleic acids that make up DNA. These antibiotics, such as sulfonamides and trimethoprim, prevent conversion of PABA to folate and therefore prevent proper DNA synthesis, you can say. Humans do not synthesize folic acid, and so these antibiotics are okay to take for humans and are selective towards bacteria. I hope you enjoyed this short video on antibiotics. Thank you for watching.
Magdi.
Then we are going to the unit 7. You are done up to unit 6, right? Right?
Yes, ma'am.
Okay, so we are going to run the unit 7 and then probably you can get some time for the review, okay? Okay, we'll do half and half if needed. Okay, okay.
Thank you.
Okay, sorry. Okay. Stop sharing. Ayan. Are you sleeping? Am I bothering you? No, no. I have a request. Actually, I am in Stark. My husband is not here. I have to take my daughter to a doctor. This Saturday, are you free to do my L8? Yeah. Uh, okay. Thank you. Okay. I can hear this is moon. I need a big favor. If possible, I'll be really grateful. If you kindly do my this Saturday LD8, just let me know because I don't have any time left. So LD8 because I have to take my daughter to a doctor's appointment. So this Saturday LD8, then I can pick up some shifts from you. I'm going to send you the WhatsApp message. Please check that one and let me know, okay? Otherwise, I have to send an email to Taylor. Thank you, sweetie. If you can help, it's going to be great help. Thank you very much. Oh dear, I'm sorry to bother you. Sorry to bother. Are you? Okay. Are you sleeping? Oh, okay. I need a favor from you if it is possible at all because I'm stuck. My husband is not here and I have to take my daughter to a doctor. Can you do this Saturday LD8 for me?
Bye, everyone. See you tomorrow.
Bye, guys. See you
See you tomorrow.
tomorrow.
Take care. Bye. See you tomorrow.